Start recording this. Okay, so today's lab, we're going to be talking about the gymnosperms. These are personally my favorite group of plants. I study gymnosperms, specifically pine species, for my master's thesis, so I'm pretty impartial to this. So just a quick review of basal land plants, which were the non-vascular and the seedless vascular plants. So with these two groups of land plants, they mainly relied on spores for dispersal. So these spores that were produced from the diploid sporophyte, they had to land in water in order for that spore to undergo mitosis and produce a brand new gametophyte. Without water or without a pretty moist environment, they wouldn't be able to reproduce at all or complete the life cycle essentially. So spores were essentially dormant until water allowed them to germinate and produce a brand new gametophyte. So remember that specialized gametangia would be produced to produce these gametes. So for the seedless vascular plants and the non-vascular land plants, those gametangia were the archegonia, which was the female, and then the antheridia, which was the male. So the archegonia obviously housed the egg, and the antheridia housed the sperm. The egg and the sperm would come together, they would undergo fertilization, and then they would produce a embryo, a diploid embryo. Then that diploid embryo would obviously undergo meiosis pretty quickly, produce the new diploid sporophyte. That diploid sporophyte would undergo meiosis and produce haploid spores. So remember that even though the sporophyte is diploid, it's going to be producing haploid spores, because then that haploid spore is going to produce a haploid gametophyte. So in order for any of this to happen, those spores have to be in water or some kind of really moist and almost muggy habitat for the free living gametophyte to grow and reproduce. So in order to complete that life cycle, again, that spore has to be in water. So this is about what the environment most likely looked like millions of years ago around the time of the Ordovician. Again, there had to be a large body of water or really just some kind of moist environment. We have some moss over here to the left. And then again, this is another image of what it possibly looked like during the Ordovician. Again, some kind of body of water. Most of the plants were pretty small. I mean, there were some seedless vascular plants like the lycopods, or certain members of the Lycopodiophyta that got really, really big. But for the most part, the plants were not that tall. Obviously, the seedless vascular plants now had xylem and phloem, so they were able to get taller than the non-vascular land plants, but for the most part, the vegetation didn't get that big. So this is the phylogenetic tree of the groups that we're going to be looking at. So over here on the left, we have the free sporing basal land plants. All the way here on the left side is the liverworts. So you guys know the liverworts as Marchantiophyta. Hepatophyta is the old name for the liverworts. We now refer to them again as Marchantiophyta, but if you see Hepatophyta anywhere, it's just the old name for the liverworts, but same group. And then we have the Bryophyta, which are the mosses. So again, these two groups together make up the non-vascular land plants. And then for the seedless vascular land plants, we have the Lycopodiophyta, which remember that included the lycopods and Selaginella. And then we have Pteridophyta, or this is actually the old name for the manilophytes. So this is basically where you would find your leptosporangiate ferns, aquatic ferns, the horsetails, whisk ferns, etc. So anytime you see Pteridophyta, or Pteridiophyta, it's the old name for the manilophytes, but it's referring to the same exact group. But now we refer to them as Manilophyta. So the name that you see in your lab manual is the more current name. So again, these are the basal land plants. That includes the non-vascular and the seedless vascular land plants. Today, we're mainly going to be focusing on the gymnosperms, also known as the naked seed plants, which are the Psychedophyta, the Ginkgophyta, and the Coniferophyta. Next week, we're going to be looking at the more derived group of species. The more derived group of species, which is the ones that you're most likely more familiar with, which is the angiosperms. Now, in between the evolution of the basal land plants and the naked seed plants, or just really seed plants in general, 4 characteristics evolved that allowed the gymnosperms and the angiosperms to really outcompete all of the other groups. So those 4 characteristics are seeds, pollen, and actually seeds and pollen evolved at around the same time. And with the evolution of seeds and pollen, it basically made spores obsolete. And then we also had the evolution of secondary xylem and phloem, and then the gametophyte being housed on the sporophyte. So now we are going into plants that basically are sporophyte dominant through and through. So the gametophyte is a lot smaller, it's a lot less complex, and now the sporophyte has kind of taken over. So the gametophyte is really, really reduced with these groups that we're going to be looking at now. So again, like I said, the three phyla, the three extant phyla at least that we're going to be looking at within the gymnosperms is Psychedophyta, Ginkgophyta, and Coniferophyta. So those are the three that we're going to be focusing on today. Just a little bit of background information on the gymnosperms. They are a monophyletic group, meaning it includes its ancestor and all of its descendants. So unlike the seedless vascular plants, those were a paraphyletic group, but gymnosperms are monophyletic. So some developments in evolution, I just went over this with the phylogenetic tree, but remember that in between the evolution of seedless vascular plants and the gymnosperms, these four characteristics evolved that really allowed gymnosperms and angiosperms to outcompete everything else. So again, one of them is the gametophyte being housed on the sporophyte. So now the gametophyte completely relies on the sporophyte. It is no longer free living, and the sporophyte is the dominant generation. So again, there was also the evolution of the seed and pollen. Those happened pretty much simultaneously. And then also the evolution of secondary xylem and phloem. So even though with seedless vascular plants, they did have true vascular tissues, it wasn't secondary, meaning they weren't producing true wood or true bark yet. And then the reason why gymnosperms are called gymnosperms is because they're essentially naked seeds, so they don't produce fruit, basically. You don't see the evolution of fruit or really flowers until angiosperms, so that's much later down the line. So no fruit, meaning that they don't have any fleshy sporophyte tissue surrounding the seeds. So if you look at, for example, the cones of conifers, their seeds are pretty much exposed and they're kind of just hiding within the cone. They don't have any fleshy tissue surrounding it. So the fleshy tissue that you eat from fruit is actually the part that you eat, which gymnosperms do not have. So that's why they're called naked seeds. So all gymnosperms evolved from heterosporous plant ancestors, meaning that they evolved from ancestors that had two separate spores. So one female spore, which is also called a megaspore, and one microspore or a male spore. So anytime you see the prefix mega, whether it's megaspore or megagametophyte or megasporophyll, that's referring to the female. Anytime you see the prefix micro, so microspore or microgametophyte, etc., that is referring to the male. So micro, male, mega, female. They also evolved from ancestors that had secondary tissues, such as secondary xylem and phloem. So secondary xylem and phloem is what really, really allow these plants to succeed, because it allowed these plants to get a lot taller, a lot larger, and because of that, they were able to resist bending and damage and grow much taller than the seedless vascular plants and the non-vascular plants. And again, because they were able to grow taller, they were able to get light a lot better than the other smaller species, and obviously resources as well. And because they're taller, they can disperse their seeds farther away, increasing their chances of reproduction, and this allowed them to be longer lived as well. So in terms of their alternation of generations, they are sporophyte dominant, meaning that they will have two copies of the genome for most of their life cycle. So sporophyte dominant, the gametophyte is completely reduced and is housed on the sporophyte, specifically in the strobili or the cones of these groups. So because the gametophyte is completely reduced, it allowed the plants to survive in drier conditions. The seed no longer requires water or some kind of moist habitat in order for it to germinate and produce a brand new plant. Whereas with the spores from seedless vascular and non-vascular plants, in order for that life cycle to continue and produce a new gametophyte, the spores had to land in water. In this case, seeds and pollen no longer need that. So this really allowed them to survive in those drier habitats. So again, they no longer depend on water, which means they can disperse a lot more inland and at higher elevations, which is a big advantage. So again, now the part that's actually dispersing is the seeds and the pollen. It is no longer the spores. Okay, so now we're gonna go into the microstrobili, or the male cones. Once again, anytime you see the prefix micro, it's referring to the male, and anytime you see the prefix mega, it is referring to the female. So the male strobilus, or the male cone, again also known as the microsporangia cones, this is what they look like anatomically, but the microsporangia are housed on microsporophylls. So I know it's kind of a lot, but remember that sporophyll is essentially the modified leaf. So again, anytime you see phyll, it is referring to a leaf. Sporophyll is a modified leaf that houses the sporangia. And a microsporophyll is the modified leaf that houses the male sporangia specifically. Does everybody get that? Is that it's a little weird, but does that make sense? Yeah, thumbs up for your video if it makes sense. And then in the chat, if you guys have any questions, just let me know. But essentially remember that a sporophyll is a modified leaf. It's a modified leaf that will house the sporangia. And anytime you just see that prefix, they throw on that prefix micro, it's just specifically referring to the male structure. So within these microsporangia, which are these circular pockets right here in that bracket, within that microsporangia is where the microspores are. So that is essentially where meiosis is going to take place to produce those haploid microspores. Eventually, those male haploid microspores are going to germinate into your microgametophyte, also known as your pollen grain or your male gametophyte. So if you zoom in, so if you look at this like axis right here, let me move this. If you look at this axis right here coming off, the actual scales are the microsporophyll. And then right here in this bracket is the microsporangium. So that's where all the haploid microspores are going to be housed. So if you look closely, I have a zoomed up image in the next slide, but if you look closely, all of these like blue little dots, those are essentially the microspores, the haploid microspores. Eventually, those haploid microspores are going to germinate, and they're going to produce the pollen grains, or the male gametophyte. So if you go on this next slide right here, so again, these scales are the microsporophylls. Again, it's a modified leaf that houses the sporangium. This entire circle right here, this entire structure is the sporangium. So again, that is where meiosis is going to take place and where all the haploid microspores are housed. Eventually, these tiny little circles, these microspores, are going to germinate and become the pollen grain, also known as the male gametophyte. So this is just a zoomed in image right here. This is just the axis of the cone, so pretty much just a ton of gametophyte tissue, or sorry, sporophyll tissue. Does that make a little bit more sense? Yeah? Okay, so once again, the microsporophyll is the modified leaf, specifically for the male, the modified leaf that houses the microsporangium. So that's this entire structure right here, this entire circular structure. Each of these individual little circles is a haploid microspore, and then eventually those haploid microspores will become the pollen grain. So that's what's going to be dispersed in the wind to allow for the life cycle to complete, essentially. Okay, so like I said, within the microsporangium are the microspores, but eventually those will germinate and produce your pollen grain, also known as your male gametophyte. Now, the pollen grain is made up of two cells. You have your generative cell, which will produce your two sperm or your two male gametes, and then you have your tube cell, which will eventually generate the pollen tube. And I'll talk about the function of the pollen tube in a second. But the pollen grain, once again, is made up of two cells, the generative cell and the tube cell. So when you think of the generative cell, just think that it generates two sperm, and then the tube cell obviously produces the pollen tube. So that's a good way to kind of remember what the functions of each of those cells are within the pollen grain. So the pollen grain was essential because it essentially provided protection for the male gametes. And with the pollen grain, because it can be dispersed in the wind, it no longer needed water for dispersal. So again, that was a big advantage over spores that were used for dispersal in seedless vascular plants. So again, the pollen grain is made up of two cells. We have the tube cell, which produces the pollen tube, and then the generative cell, which produces two sperm. Now, even though it produces 2 sperm, it'll typically fertilize both of the eggs in the female cone, but usually only one of them will survive. Okay, so like I said, the pollen tube, which is produced from the tube cell of the pollen grain, what it basically does is when that pollen grain, when that microsporangium opens and disperses all the pollen grains through the wind, hopefully that pollen grain will land on a female cone or a female ovule specifically. On that female ovule, when that pollen grain lands, what it's basically going to do is that it's going to produce, or it's going to get a cue to produce the pollen tube. And that pollen tube is pretty much going to enter the embryo sac into the egg, and then it's going to burst and release the sperm, and then that will essentially fertilize and then produce your new zygote. So again, the pollen tube pretty much digests the path to the egg. It's going to locate the micropyle, which is basically just a small opening on the surface of the female cone. And then once it lands on that micropyle, once that pollen grain lands on the micropyle, the tube cell will get a cue to produce the pollen tube, and then the sperm is going to get released and it's going to fertilize the egg essentially. So this is the pollen tube. Again, that's generated from the tube cell, and then these are the 2 sperm cells that were produced from the generative cell. This will eventually burst and then it'll fertilize the eggs as soon as it lands on the micropyle. So now we're going to move into the female strobili, or the female cone, also known as the megasporangiate cones. So the it's the same thing where with the males, you had the microsporophyll that housed the microsporangium. Same thing for the female, where the megasporophyll, which again is the modified leaf, but now we're specifically referring to the female, houses the megasporangia, or the female sporangia. So this is what the anatomy of a female cone looks like. These are the ovuliferous scales. So these are essentially these big scales coming off right here. These are basically the megasporophylls. At the very end of those megasporophylls, right here is your megasporangia, or your ovule. Now your megasporangia is where your megaspore is going to be housed, your haploid megaspore. So then if you zoom in right here on this animation, on this part right here, here is the integument. This is part of the ovule, or the megasporangia. Basically what that is protection for the ovule, protection for the megaspore. Eventually, this integument is going to become the seed coat once the seed is fully fertilized. So this is your integument. Inside would be your megasporangium, and then right here in the center would be your haploid megaspore. The entire structure, though, is called the ovule, and the ovule is going to be right here at the very end of the megasporophyll. So again, these scales are the megasporophylls. Here would be your ovule. If you zoom in on that ovule, this specifically, the outermost region of it would be the integument. Again, that just provides protection. And then inside would be your megasporangium. Again, your megasporangium is where your megaspores are housed, which is right here in the center, your haploid megaspore. Okay, so if we look at this, these drawings again right here, once again, this is your megasporophyll, which is essentially the scales of the cone. And then at the very end, within that megasporophyll would be your ovule. So this right here is the micropyle that the pollen grain essentially has to find when it's dispersed in the wind. And hopefully that pollen grain lands right on the micropyle, and then the pollen grain, specifically the tube cell, will get a hormonal cue to produce the pollen tube, and then the pollen tube is pretty much going to grow right into the ovule, enter the embryo sac in the egg, which is right here, and then it'll fertilize those 2 eggs. Because remember, it produces 2 sperm. With the female ovule, it'll produce 2 eggs, but usually only one of the zygotes, or one of the embryos, will survive. Okay, so with the female cones, you have a diploid megaspore mother cell that's going to go through meiosis and produce 4 haploid megaspores. That's all happening within the megasporangium. So remember, the megasporangium is where meiosis takes place just like how with the male cones in the microsporangia, that's also where meiosis takes place and produces your haploid microspores. Same exact thing with the females, where your megasporangium, which again is found within the ovule right here, will produce your haploid megaspores. Now, specifically for the females, it'll produce 4 haploid megaspores, but only one of them survives. Only one haploid megaspore survives to produce your haploid megagametophyte, which is this right here, to produce your female gametes or your eggs, which is right here, archegonia with the eggs. So this is one archegonia with one egg. This is the second archegonia with the other egg, all within the female gametophyte, which is right here. So this entire structure is the ovule, and it's found within the female cone at the end of the megasporophyll. Does anybody want me to go over that again? Anybody want to unmute themselves or write in the chat? Okay. Oh wait, there's 2 messages. Okay. Seems like you guys are good, so I'm gonna move on. Oops. Okay, so right here is basically how pollination occurs in this image right here. So this animation right here is a photo kind of zoomed in on the microsporangium, which again is the male sporangium. Now this microsporangium already produced the haploid microspores, and then those haploid microspores spores essentially germinated and produced pollen grains. So that's what these individual circles are. These are the pollen grains. Eventually that microsporangium is going to open, is basically the cone is going to break apart, and the pollen grain is going to get dispersed in the wind. When it gets dispersed in the wind, hopefully it'll land on a female cone, specifically the integument, which is, or is really the micropyle of the integument. So the micropyle again is just the opening on that integument or the ovule. So this is the integument right here. So that pollen grain will land right there on the micropyle. Then the tube cell of the pollen grain is eventually going to get a hormonal cue to produce a pollen tube. That pollen tube is then going to grow down into the embryo sac. It's going to burst to release the 2 sperm, and then it'll fertilize the eggs, and then eventually producing a new embryo. So again, like what it basically says here, male gametophyte sends down a tube, that's specifically the pollen tube, to infiltrate the ovule and send down its sperm into the egg. So if you look right here to the right, this is essentially what it'll look like. So once that pollen grain landed on the micropyle, it'll produce the pollen tube, it'll burst, release the sperm, and then it'll fertilize the egg. Once again, even though it produces 2 sperm and 2 egg, usually only one of them will survive, and only one embryo will live basically. Okay, so with cones, in order for this to get dispersed, they have to be open. So this is pretty much what a closed cone would look like. If a pollen grain were to land on a closed cone, pretty much no fertilization would occur because the seed is inside, like at the very end of the scale. So here's the cone scale right here. This is specifically a female cone. So this is the ovuliferous or the megasporophyll. And then at the end would be your seeds. Eventually though, when the cones open, so when they open, that's where the seed is right there, then fertilization can take place. But some cones are really, really stubborn. They won't open unless they're exposed to really, really high temperatures. Like one of the species that I'm studying, which is Pinus attenuata, they won't open their cones unless there's a fire. So reproduction is usually pretty low. Okay, so now we're specifically going to be looking at the seed. There are 3 different generations for the seed. That's the main thing I need you to get out of this slide. 2 of them are diploid, 1 of them is haploid. Now the integument, which eventually becomes the seed coat for a mature seed, is diploid. So again, this just essentially provides protection for the embryo. The megagametophyte or the female gametophyte acts as the food supply or like the nutritive tissue for the embryo. Now the female gametophyte, that's haploid, but the embryo, obviously that's going to be diploid because in order for the embryo to be produced, a haploid female and a haploid male had to come together and fertilize and produce this brand new embryo. So the embryo and the seed coat of a mature seed are diploid, whereas the female gametophyte or the food tissue is haploid. And this embryo, again, is taking all of its food and nutrients from the female gametophyte. So the seed coat, which again was the integument, essentially allows the embryo to go dormant. It's protecting it from certain environmental conditions, but when the environmental conditions are good or favorable for germination, then the embryo will essentially not go dormant anymore. But again, that seed coat is basically providing protection. Okay, so now we're gonna go into the specific phyla. The first one that we're gonna be looking at is Cycadophyta. These are really, really cool. You've probably seen them growing in your neighbor's yard because a lot of people like to plant these in landscaping. So the Cycadophyta, which is the more basal group that we're going to be looking at, these were dominant during the Mesozoic. We usually refer to them as the early gymnosperms. They do have monopodial growth. So now we're moving away from species that had dichotomous branching, which is what we saw a lot in seedless vascular plants like Selaginella. So with monopodial growth, they have one axis or one main stem that doesn't branch, or it really doesn't branch often at least, but it'll pretty much branch from that one main stem. So with cycads, they have a really woody stem with a really wide pith. So they do obviously produce secondary xylem, but actually most of the volume of their stem is actually that really wide pith. They are evergreen, meaning that they don't drop their leaves during certain seasons. They'll keep it for the entire their entire life cycle. But they have really stiff pinnately compound leaves that contain tons of lignin. So remember, lignin is that chemical that provides rigidity in the cell walls and pretty much keeps it nice and hard and stiff. And they do have a lot of lignin in their leaves because they need to last a long time. Keep in mind, they are an evergreen species. And this also helps prevent damage to tissues if they get dehydrated. They evolve during a really dry period, so the lignin essentially allows them to resist cavitation, or essentially the air bubbles within the xylem. So they do have flagellated sperm. Their pollen will essentially send down a haustorial structure or tube that'll burst when sperm swim down to find the egg. But their sperm is still flagellated. So they are mostly dioecious, meaning that they have separate male and female plants, meaning they're going to produce a separate male spore and a separate female spore. So di meaning two, ecious meaning house, two houses, two separate plants. So there are 4 different genera or genuses that we're going to be looking at within Cycadophyta. We have Cycas, and then we have Zamia, Encephalartos, and Dioun. We also have tons of pictures of each of these genuses within the PowerPoint that you guys are going to use for the lab. So Cycas, these are usually the ones that most people can recognize easily because they have really, really thin leaflets and they have only one single vein going down each of those leaflets. So if you look closely at a Cycas, you'll see that there's kind of like one indentation going down the center of that leaflet. That's going to be the single vein. So as soon as you see really thin leaflets with one vein going down the center, you should know right away that that's a Cycas. And then we have Zamia. This is personally my favorite one because they're just so cute. But Zamia usually has really smooth and like rounded edges. And when you touch a Zamia leaf, they're usually also kind of fuzzy too and really, really thick. So that's one way that you can recognize Zamia. Unfortunately, you guys can't touch it, but if you're ever on campus, when this is all over, go to the Mesozoic garden. You should be able to spot Zamia really easily, and you should know it by the feel of their leaves as well. But these are going to be also pretty easy to tell apart because this is the one that has the rounded and kind of smooth edges, whereas Encephalartos, their leaflets are a lot wider, but they have spines on the margins of their leaflets. So you can kind of see in this image right here. So that's a really good way to tell the difference between Encephalartos, Zamia, and Cycas. They all kind of have their own characteristic that sets them apart. Now, Dioun is the one that's a little bit harder to recognize because sometimes it'll kind of look like Encephalartos, but for the most part, although they do have spines on the edges of their leaflets too, just like Encephalartos, usually the leaflets for Dioun are going to be much narrower and thinner in comparison to Encephalartos. Encephalartos, usually their leaflets are going to be wider. But the two that are definitely easy to tell apart is Cycas and Zamia. So remember that Zamia has the smooth, rounded leaf edges, whereas Cycas has that one vascular bundle one vein going down the center of the leaflet. Sagus palm leaves, but is it? No, it's not. It kind of looks like it, but it's not. Yeah. Okay, so now for the cones of the Psychedophyta. Now the male cones are usually going to be woody and persistent, meaning that they're going to last a long time. They do get pretty large, but they're nowhere near as large as the female cones. And then the female cones, they're going to be larger in terms of volume at least, but they are ephemeral. So when I use the term ephemeral, what that means is that eventually those cones are going to crumble apart over time so that they can disperse the seeds or the pollen. So right here on this photo in the center that my cursor is on, this is a female cone right here. It is huge. Female cones of cycads are ginormous, but eventually, these female cones are going to open and break apart, which it'll look something like this when they start to break apart so that they can disperse their really colorful seeds. This right here at the top is what a male cone looks like. So this is, the male cones are usually a lot longer. Right here in the center is a male cone that's starting to develop. But yeah, so male cones are woody and persistent, meaning that they don't break apart, whereas the female cones are a lot larger, but they will break apart over time. And again, it's because they're going to be dispersing their seeds. Now, sometimes the female cones can exude nectar to attract pollinators as well, because for the most part, cycads are actually mainly dispersed by animals. And that's actually why their cones are usually a lot brighter and a lot more colorful, and that's to attract vertebrate animals. And that's actually also why their seeds are usually pretty colorful too. That's to attract certain animals. When cycads evolved, it was actually a lot more advantageous to have animal pollination because they were mainly found in really densely vegetated tropical forests. So it really didn't make sense for their seeds to be dispersed in the wind. So instead, animals would take their seeds and disperse them a lot farther into the forest compared to if it was just the wind. So again, that's why their cones and their seeds are a lot more bright and colorful. So these are examples of some female cones right here. This plant is actually in the Mesozoic garden. I actually took this picture. And then these are another example of some female cones of cycads as well. Okay, so the next phyla that we're going to be looking at is the Ginkgophyta. There is only one species left in this phylum. In this entire phylum, there's only one species left, and that is Ginkgo biloba. This phyla actually used to be pretty diverse, but unfortunately now there's only one left. I'm sure many of you guys are aware of Ginkgo biloba because a lot of people use these as like supplements, I think, for digestion, something like that. I know it's a supplement. Just like Psychedophyta, they are dioecious, so they have separate male and female plants. The female plants produce these horrid smelling seeds. You guys actually kind of lucked out with this because usually we'll bring in the ginkgo seeds in the lab and they're like in this kind of sketchy looking Ziploc bag. And I always tell my students, don't open the bag. And somebody always opens the bag and it smells. So you guys kind of lucked out on this one. You don't have to smell it this semester. But they are deciduous, so unlike Psychedophyta, Psychedophyta was evergreen, meaning that they'll keep their leaves on for the entirety of the season. Ginkgos will drop their leaves specifically during the winter, and that's pretty much to eliminate the chances of them getting freeze-induced cavitation. But just like cycads, they do have flagellated sperm, so again, that just means that their pollen is going to send out that tube. But so characteristics that they have in common with cycads is that they're dioecious and that they have flagellated sperm, but they are deciduous, whereas cycads were evergreen. So ginkgos are really cool. They have dimorphic leaves, meaning they have two different morphologies essentially. They have long shoot leaves and short shoot leaves. Now the long shoot leaves, which I have right here at the top, these leaves are usually going to have a much deeper lobing, which you can also see in these images right here. Whereas the short shoot leaves, which you can see in this photo right here on the top right, they're usually going to be more entire. When you use the word entire, that pretty much means that the edge of their leaf is smooth. So again, short shoot leaves, they have more of that smooth edge, whereas long shoot leaves, they have more of that deeper lobing. But for the most part, they're kind of like fan shaped, where they kind of like widen out. They do have dichotomously branched veins though. So pretty much like what that means is that their veins like with dichotomous branching with Selaginella, where it starts off as one and then goes into two and then two and then two. That's what the vascular bundles or the veins of the leaves will do for ginkgos. But again, main thing is that long shoot leaves have that deeper lobing. Short shoot leaves are more smooth or entire. Now the cones for the ginkgos, both female and male, they're pretty small. Both female and male are pretty small in comparison to cycads, where the cycad cones, whether it's female or male, were huge. But both the female cones and the male cones are typically going to be found on the short shoot leaves. And the female cones will usually release a droplet of nectar, which you can see in this image right here, at the very tip to attract pollinators, just like how the female cones of cycads release nectar as well to attract animal pollinators. But if you look at this image right over here where the short shoot is, let me move this. So this is a short shoot right here. You can think of them as like a tiny little lateral branch. And remember, the leaves that come out of the short shoot are going to be a lot smoother. So again, that is where your male cones and your female cones are typically going to be found. Okay, so now we're going to talk about the horrid smelling seeds of the ginkgos. So the seed really does look like a fruit, and that's because it has a fleshy or a fleshy seed coat, but it is not a fruit. Fruit has not evolved yet. We're going to be talking about fruit next week when we go over angiosperms. But their seed coat has 3 layers. They have 2 hard layers and 1 fleshy outer layer. And that fleshy outer layer is what made people think that they're fruit, but they're actually really just seeds with a really fleshy seed coat. So remember that there are 3 different generations within the seed coat. You have your within the seed. You have your diploid embryo, your diploid seed coat, which again derived from the integument, and then you have your nutritive tissue or your female gametophyte that supplies the nutrients and the food to the embryo. And that nutritive tissue is haploid. Okay, now we're gonna go into the Coniferophyta, my personal favorite. This is the largest phylum within the gymnosperms. This is pretty much what you're probably what you've been more exposed to. So this includes pines, redwoods, also a few new ones that you probably haven't heard of. Also includes Podocarpus and Araucaria, and the really weird but really, really cool Nitales. So within the Coniferophyta, unlike cycads and ginkgos, these guys are monoecious, meaning that they have their male and female cones or their male and female gametophytes on the same plant. They're all housed on one plant. However, some southern hemisphere conifers, such as Araucaria and the Nitales, actually, those are all going to be dioecious. So those are going to have a separate female plant and a separate male plant. But at least the conifers that you guys are exposed to here in California that are native to California, those are going to be monoecious. So with the conifers, their male and female cones kind of switch compared to the cycads, whereas with the cycads, the male cones were woody and persistent and the female cones were ephemeral. It's the opposite for conifers. The male cones of conifers, which is right here on the left, they're really, really small, tiny, and they're ephemeral, meaning that those cones, if you touch the cones, they'll basically crumble apart, and that's basically them dispersing their pollen. And that's why they have to be really small and ephemeral, because that pollen is wind pollinated. So it's pretty much advantageous for them. The female cones, which is right here on the right, those are usually going to be a lot larger, they're going to be lignified, and they're going to be long lasting as well. So for the conifers, the female cones are much larger and they're persistent, whereas the male cones are tiny and ephemeral. And the male cones have to be small, they have to be able to easily crumble apart so that they can disperse their pollen. So these are some more examples of some male cones. Now some male cones, depending on the species, can be longer than others, but for the most part, they are pretty small in comparison to the female cones. These are some examples of female cones right here. These 2 species right here on the far left are the 2 that I study. Right here on the top is the coulter pine, Pinus coulteri, also known as the widowmaker. These cones can get huge, and the reason why they're called the widowmaker is because they have these really sharp talons at the end of their scales, and if a cone were to land on your head, you'd basically be dead. So that's why they're called widowmakers. They're huge. They're ginormous, and they can get pretty heavy as well. And then this is the other example of a female cone, but this is a species, the second species that I study. This is Pinus attenuata, or knob cone pine. Now this is a pine or cone that is an example of a serotinous cone, so they will not open their cones unless there's a fire, essentially. So they need those really, really hot temperatures in order for their cones to open. And then we have sugar pine right here, Pinus lambertiana, and then we have actually the coast redwood, Sequoia sempervirens. This is what their female cones look like. Okay, so with the conifers, they have pollen grains that essentially develop bladders. So that's these Mickey Mouse ears right here that I have with the arrows that basically help them float on the air and travel farther. So when those male cones crumble apart, what helps that pollen get dispersed in the wind are these bladders, so these Mickey Mouse ears. So unlike cycads and ginkgos, the sperm is not flagellated, so it requires that pollen tube to reach the egg. Remember that pollen tube is produced from the tube cell of the pollen grain. So right here, you can't really see it that well, but eventually this will have a nice and clear tube cell here, or sorry, tube cell here at the bottom, and then your generative cell here at the top. That tube cell will produce your pollen tube, which is essentially this right here, what my arrow and my cursor is on. So again, once that lands on the micropyle of the integument, then that the tube cell, which is that right here, will produce this pollen tube. And then the generative cell will produce 2 sperm, but 1 sperm will fertilize the egg, the other one will die. Okay, and then finally, a group within the conifers is the Natales. The Natales were actually thought to once be more closely related to the angiosperms, and the reason why they were thought to be really closely related to the angiosperms is because they have vessels. Now keep in mind, non-flowering plants, so all the gymnosperms that we were talking about just now, cycads, ginkgos, and conifers, they all only had tracheids for water transport. They did not have vessels, which is why the Natales were once thought to be the sister group or the sister lineage to the angiosperms, the flowering plants, because they have vessels, but it turns out they're actually nestled within the conifers. So they actually also have double fertilization as well, which results in the production of 2 embryos. So actually both sperm are going to be used to fertilize those 2 eggs. One of them is eventually going to die, but that is one characteristic that angiosperms have as well, double fertilization. So double fertilization and vessels were a result of convergent evolution, meaning that they basically evolved those traits separately, even though they don't have a similar common ancestor. So again, the Natales are a group within the conifers. They also pretty much look like angiosperms for the most part. Sometimes their cones look like an inflorescence or a cluster of flowers, which is also why they were thought to be an angiosperm, but they're actually a group within the conifers, so they are a gymnosperm. The 3 species that we're going to be looking at are Welwitschia. These are native to the Namibian desert in Africa. These are crazy because throughout its entire lifetime, they only produce 2 leaves, 2 freaking leaves. So they have a basal meristem with wood. Now, if I show you guys what Welwitschia, I'm just going to show it to you right now because why not? This is Welwitschia. That is 2 leaves. It gets crazy big. I included in the Lab 11 slides that you guys are going to download to answer the questions. I have some other pretty crazy pictures of Welwitschia, but yeah, this is basically 1 leaf, and this is basically the 2nd leaf. I know it looks like multiple leaves because it starts to get tears in the leaves as it gets older, but that's 2 leaves. This is where the basal meristem is, and the basal meristem can actually get woody too, it's just insane. This plant is weird. But then we also have Ephedra. Now, Ephedra has really obvious vessels. I mean, all the Natales have vessels, but the Ephedra just makes it a lot nicer. But they have smaller cones in comparison to Welwitschia, and the female cones of Ephedra will basically only develop 1 seed. And then we have Nita. Now these guys really look like an angiosperm. They have really broad leaves, and they actually have pinnately compound leaves with nice net-like venation, and they really do look like angiosperms. But once again, this is Welwitschia. And then over here we have Ephedra. Ephedra actually kind of reminds me a little bit of Equisetum or the horsetails from the seedless vascular plants, but they are not. They do produce cones at the very tip as well. This is what Needham looks like, the whole entire tree. And then these are the cones of Needham. But I have some other good photos as well in the Lab 11 slides. But yes, so Needham, Ephedra, and Welwitschia, all within the Natales. They do have vessels for water transport, but remember that they are a conifer, so they are a gymnosperm. They are not angiosperms. Even though they have similar characteristics to angiosperms, they are not angiosperms. Okay, so that was it for my lecture. I love this lab a lot because I love gymnosperms, but that was pretty much it. So remember to take your pre-lab quizzes for Lab 11. That should have been live on Blackboard already. I know that some of you guys already took it. Awesome. That quiz is going to close at 11 a.m. So please remember to take the quiz. It's really, really simple and straightforward. It's only 5 questions. You have 7 minutes to take it, but it's probably not going to take you the full 7 minutes. What else? So next week is Lab 12, which is angiosperms. That's going to be the last lab. Woohoo, the last lab, and then you guys are free of me. Not quite, you still have to take your final practical. But just to go over some dates really quick. So next week is Lab 12, angiosperms. That's also when you're going to be turning in the lab assignment for Lab 11, which is this lab. The week after, which let me just open my calendar really quick to make sure I get the dates right. So yeah, 28th is going to be Lab 12, also when Lab 11 is due. May 5th is when you are taking your final practical and when your final paper is due. And when Lab 12 is due, actually. So you have a lot going on that day. If you guys feel like it's too much, I can go ahead and move some days around. But as of right now, May 5th is when Lab 12 is due, when your final practical is, and when your final paper is due. That's a lot, I know. So there's a chance that I actually might be moving some days around. But as of right now, just keep in, what will definitely stay is your final practical. So your final practical will definitely be May 5th. And you will also turn in your Lab 12 assignment that day. I might move your scientific paper deadline to a couple days after that. Actually, yeah, let's do that because you have your final practical that day. So, okay, now I'm gonna make your paper due on May 8th. Friday, May 8th is the absolute last day to turn in your paper. Does that sound good to everybody? Thumbs up. Okay, so Friday, May 8th is when your scientific paper is going to be due to Blackboard via Turnitin. So I can see if you guys are going to be cheating or not, but don't do it. You guys are adults. Don't cheat. Okay. Also, so Friday, May 8th. So that means that the last day that I'm going to accept your papers for me to review, like if you want to just email it to me and have me go over it really quick before you actually submit it, the last day for that is going to be Monday, May 4th. So if you want me to read it, look it over, give you a thumbs up before you turn it in on Blackboard, it's going to be Monday, May 4th, where you can email that to me. Does that sound good? Okay, so Monday, May 4th, and then again, final practical in Lab 12 will be due May 5th, right? Okay, well, if you guys don't have any other questions, you're pretty much free to go. This lecture has already been posted to your Blackboard. I also emailed it to you guys. Everything should be pretty much up to date on our Blackboard. Once this is done, once we're done recording this and stuff and it's converted to an MP4, I'm going to upload this to YouTube and then I'll send you guys out the link later today as well. But yeah. Okay, so you guys are free to go. See you next week. Have a good rest of your week. Good luck on your lecture exam. I'm sure it'll be really hard, but hopefully you guys studied. I'm just telling you the truth. It'll probably be hard. Okay. Well, if you studied, it shouldn't be that bad. But okay, listen, when I was in undergrad and I was taking this class, I thought this class was super hard. And then looking back, it really wasn't. You just got to study. Okay, so I will be here till, what time is it? Yeah, I'll be here till about 11 o'clock when the class is supposed to officially end on Zoom if you guys have any questions. So feel free to log off and then log back on if you guys have anything that you need to ask me. So yeah, let me stop this share. an online party instead of a final? We can do it after the final. Okay, let's stop this.